I'm Keely Rasha, and this is my project, Silver Slaughter. Here's my abstract, safety sheet, and then here are my 2 endorsements. Acknowledgements. I would like to thank Ms. O'Dowd for purchasing the materials that I couldn't and for assisting me in my testing. I would like to thank my mom, Ann Felder, and my dad, Scott Rasha, for purchasing the materials and supporting me in my endeavors. Finally, I would like to thank Ethan Ammerman for his emotional support. Purpose. The purpose of this project is to figure out the least amount of colloidal silver that creates a zone of inhibition in E. coli. My independent variable is the amount of colloidal silver applied. The specific amounts of colloidal silver are 500,000 micrograms per liter, 50,000 micrograms per liter, 5,000 micrograms per liter, 500 micrograms per liter, 50 micrograms per liter, and 0 micrograms per liter. My dependent variable is the zone of inhibition. 4 of my constants are the time that each bacteria in each trial can grow in each petri dish, the type of bacteria, the number of nutrients given to the bacteria, and the type of colloidal silver. Continued, I chose this project to help bring awareness to the different ways to kill bacteria and how effective colloidal silver truly is in eliminating bacteria. Many people use it on their bodies despite the negative side effects, and I want to see if that negative effect is worth it. I want to be able to see the exact measurement that is needed to reduce side effects and to waste the least amount of silver possible. Silver is not a renewable resource, and we need to know specifically how much silver we need and how it is to know how to create safe, efficient, and ethical products for everybody. Here's hypothesis. If the 50 micrograms per liter of colloidal silver are distributed into the bacteria, then the filter circle will have a visible inhibition zone. Since one bacteria is equal to 100 nanosilver, and silver is very deadly to bacteria in very small amounts, even silver that is together half the size of one bacterial cell will create and spread a zone of inhibition. Background research. Each of these will be developed into 5 paragraphs. This is the introduction, and each one as it goes on will have a different one. People think that the werewolf myth is only drenched in fairy tales and superstitions. However, the myth is partially based on a fact. Like werewolves, silver is deadly to bacteria. As with werewolves, the most effective and primary way to kill bacteria is with silver. Silver is found in many products. Some of the most common and known ones are socks to prevent odors, food packaging to prevent mold, and medical drugs to prevent infectious growth. They can be used for anything and everything. In microscopic amounts, silver is put into most packaging and sometimes in the object itself in the form of nanosilver. But how much silver is required to be put into that packaging? What if people get sick, or the food spoils, or the socks stink? Scientists need to know how much nanosilver exactly to put into the packaging to kill bacteria to avoid unnecessary illnesses and even death, a waste of materials, and much more. In order to understand how the amount of nanosilver affects E. coli's life, one needs to know the structure of E. coli, antibacterial substances, what colloidal silver is, its structure, how it affects bacteria, and what nanosilver is. E. coli is a gram-negative Bacillus bacterium 1 to 2 micrometers long with a 0.5 micrometer radius. The bacterium is commonly found in the lower intestines of warm-blooded animals, but it can also be found in food and uncontaminated water. It is a heterotrophic bacterium, meaning it feeds on food produced by other organisms, but it can turn autotrophic eating carbon dioxide if it doesn't have enough nutrients. There are many strains of E. coli, and they all do different things to survive. Some are harmless, some help humans, and some, when ingested, can cause serious illnesses such as food poisoning. It has a prokaryotic cell structure since it does not have a nucleus, and instead it has a nucleoid and a cell wall. E. coli also has a cytoplasm, cytoplasmic membrane, a capsule, ribosomes, a plasmid, and a flagellum. Since E. coli is a gram-negative bacterium, it has a layer of lipopolysaccharides that is 1 to 3 micrometers thick, and peptidoglycans that are 8 nanometers thick, or 4/500ths of a micrometer. Due to the thinner protective walls, it is theorized that the nanoparticles cause greater destruction to the cell wall and are easier to get into these type of cells. Colloidal silver is a mix of water and nanometer-sized pure particles. Due to its antibacterial qualities, this substance is used to kill bacteria and food products on surfaces and sometimes in the human body. In standard silver, all of the particles are bonded together tightly. With colloidal silver, though, the particles float around the water, so they don't bind to each other as often. Colloidal silver particles are spread apart, and these separated silver particles get into bacteria more quickly due to their small size. All types of silver are made up of silver ions. These ions are well known to cause death in bacteria due to restricting the bacteria's access to their DNA, thereby stopping production. blocking their access to the respiratory system, thereby destroying energy production, binding to the cell wall, thereby weakening the cell wall, stopping any nutrients from getting in and any destructive materials from getting out. Without its cell wall and membrane protecting its contents from the outside, the cell explodes, thus killing the organism. It is also noted that colloidal silver, after it kills the bacteria, it acts sponge-like. It stays with the dead bacteria, infecting living things that are left in the vicinity. The larger the amount, the more bacteria it can infect. When colloidal silver is applied to bacteria, the area around it is called the zone of inhibition, which is an area where bacteria cannot grow. Nanoparticles are particles that are between 1 to 100 nanometers in size, which is equivalent to 10 to the negative ninth power of a meter to 10 to the power of a negative seventh of a meter. 1000 nanometers equals 1 micrometer, the smallest size of an E. coli cell. They're created by the breaking down of larger particles, which is naturally done with substances by cooking or by forcing them with one of the 6 methods: electrochemical, photochemical, radiolytic, or sonochemical methods. Those methods are all called a bottom-up approach, when something crushes the material until it breaks the ions apart. Those methods break up the bond between the clusters, ions, and sometimes molecules until all that's left are the nanoparticles. This is what happens in colloids, except for the water acts as the solution to break apart the material, and it is usually less severe. It usually only breaks it up into smaller clusters, not ions. Nanoparticles of antimicrobial substances, when sought out specifically, are primarily used for types of medical care as an antibody due to their ability to get in almost everywhere. Still, they can be used as an antibacterial in materials such as socks, washing machines, doorknobs, and food. Due to their larger surface area and smaller size, they are more helpful in eliminating microorganisms than their counterparts. To waste the least amount of material in the society that is constantly growing bigger, scientists need to be aware of the minimal amount of silver they need to use in products. Silver is not a renewable resource, as I said before, which means people need to stop using it recklessly. Everyone deserves to be safe from infections, especially from E. coli, and this project can guarantee that with the least amount of excess waste. This is why, to be able to put nanosilver in most products, and to be the most accessible, one needs to be educated on the structures and life of E. coli, colloidal silver's benefits, and why nanoparticles are so essential to killing bacteria. Here are the materials that I used. Bleach, 6 small, clear, transparent jars, colloidal silver, 550 parts per million, distilled water, 60 sterile filter discs, 6 tweezers, E. coli, 2 graduated cylinders that can hold 22.5 milliliters, medicine droppers, 50 sterile Petri dishes with nutrient agar plates, sterile cotton swabs. Procedure: clean all necessary equipment and surfaces thoroughly with bleach, put the disposable gloves on, and find a flat and clean surface to test on. 2. Put the 6 mason jars in a row and label them A through F. 3. Pour 22.5 milliliters of the distilled water into the mason jars labeled B through F. Use a graduated cylinder to make sure. 4. Pour 25 milliliters of the colloidal silver solution into mason jar A using the graduated cylinder. 5. Using the medicine dropper, transport 2.5 milliliters of the colloidal silver from A to B. Use graduated cylinder to make sure it is 2.5 milliliters and squirt any extra silver that was left in the dropper back into jar 1. 6. Mix the colloidal silver in B with the distilled water. 7. Repeat steps 5 to 7, but use the previous one's mixture for each mason jar instead of jar A's. Don't put any colloidal silver in jar F. 8. Separate each Petri dish into equal quadrants. Label each quadrant as a number corresponding to the jars. Make sure there are 10 of each letter. 9. Open the E. coli container and open a Petri dish. 10. Get a sterile cotton swab and gently rub the tip of the cotton swab with the E. coli mixture about 10 times. 11. Spread the bacteria throughout the Petri dish by zigzagging through the whole plate from left to right and top to bottom. Rotate the plate and do it again a couple times. 12. Repeat step 11 until all Petri dishes have been filled with bacteria. 13. Get 6 separate plates and tweezers for each of the colloidal silver mixes. Separate the filter paper circles evenly on each plate. 14. Drop 1 to 2 drops of jar A's mixture onto a filter paper. Turn the filter paper over with the tweezers, then place it closest to the center as it can get on the quadrants marked A. 15. Repeat step 14 9 more times with jars A mixture, then repeat with the other mixtures. 16. When done, tape the Petri dishes. 17. Place the Petri dishes on a plate, then put it somewhere where it will be around the same temperature for a few days. 18. After about 4 to 7 days, get a ruler that measures in, I meant to say centimeters, a magnifying glass, and a petri dish. Go to a window or a bright space and measure one of the filter circles. Measure the space that is clear around the filter paper with the ruler. 19. Record the results in the data table. 20. Repeat the steps 18 to 19 59 times. 21. Put the petri dishes in an incubated place and repeat steps 18 to 20 after a couple of days. Repeat until the zone of inhibition stops growing. So I have a few data tables. This is number 1 for day 4. This is also test 1 until it says so. All of these will be test 1. In this one, all of the zone of inhibitions on test F were more watery, not clear like the others. I will show this later. And then here was the graph. And here was number 2. This was day number 8. The ones with Na on them had mold growth affecting the measuring of the zone of inhibition, and this applies mostly to all future tables as well. I will say when it doesn't apply. For trial 7E, there was condensation on the petri dish, so I could not accurately tell if there was a zone of inhibition or not. Here was data tables number 3, day 12. The trials that are not on this graph only had Na data, so it wasn't sufficient to put them on the table. Right now, those trials are 1, 3, 8, and 9, as noted if you want to look at it here. Also, like I said earlier, this is an example of trial F's discrepancy noted earlier. Look at F6. It was for day 12. Here was data tables for day 19. Trials 6, 7, and 8 only data was for group E, or the 50 micrograms per liter group, and it was all 0, so it does not appear on the graph. This is test 2. In this data set, more of the cereal dilution was poured onto the filter circles, plus a different method of putting the E. coli on the petri dish was used, which may be causing the discrepancy in the size of the zone of inhibition. I also only did groups C through F, which is why there isn't 50,000 micrograms and 500,000 micrograms. And here's the graph. On trials 11 through 13F, there were little opaque dots on the zone of inhibition, so the data could be inaccurate. This is still talking about day 5, both of these. And I'm also unsure about trial 11's D data due to the fact that there were 2 different rings. The same problem applies, just the zone of inhibitions were very tacky, I guess. And unlike the ones observed in the ones with more colloidal silver. Here was data tables for day 12 on test 2. On trials 12 through 14F, there were little opaque dots, as shown earlier in the zone of inhibition, so the data could be inaccurate. Here's the explanation of results. My data shows that the amount of colloidal silver does impact the zone of inhibition. The more colloidal silver per liter increases the length of the zone of inhibition around the paper filter, which means it is more effective in eliminating E. coli in greater amounts. However, my results also show that the distilled water develops a zone of inhibition as well, though it appears different from the ones developed by the colloidal silver. Overall, there is very little growth in any of these zone of inhibitions after the first 4 days. Some experimental errors that impacted my experiment are the following. I frequently had to infer the length of a zone of inhibition due to how small it was, so the measurements might be slightly inaccurate. The amount of E. coli could not be controlled, which affected the size of the zone of inhibition. There is a substantial amount of mold that impacted some, if not most, of the results recorded, which impacted how many results I got. Trials 1 to 10 use a different method of putting the bacteria on the petri dish and the cereal dilution onto the filter paper than trials 11 through 14 CDEF did. That's marked as test 2. So that is causing the discrepancy in the data noted in the table. Conclusion. So the first 2 right here will be paragraph 1, and then the second paragraph will be just one thing, and then the third paragraph will be broken up like this. And I will note that. The purpose of this project is to figure out the least amount of colloidal silver that creates a zone of inhibition in E. coli. My project concludes that 50 micrograms per liter is the lowest amount of colloidal silver needed for a zone of inhibition to appear, which sufficiently answers my purpose. If I count the zone of inhibitions encountered in my control, there isn't enough evidence to prove or disprove my hypothesis. My hypothesis read, if the 50 micrograms per liter of colloidal silver are distributed into the bacteria, then the filter circle will have a visible inhibition zone. In the experiment, most of the different subjects in E appeared to have one, which appeared to have an average of 0.025 centimeters in length after 19 days in the first test, and measured an average of 1.375 centimeters after 12 days in test 2. Since this was the lowest amount of colloidal silver used in my experiment, it is the lowest amount that needs to be used to kill bacteria. However, since the experiment also concluded that pure distilled water with no colloidal silver added produced a zone of inhibition, it is hard to prove what effect the concentration of colloidal silver had on the zone of inhibition on its own without the help of the water, so the hypothesis cannot be answered sufficiently. In the first test, disregarding the second test, evidence due to the dots noted, Group F had an average zone of inhibition length of 1.5 centimeters. After 12 days, I meant centimeters and millimeters. After 12 days, and started with an average length of 0.65 centimeters. Though it was a different appearance, I cannot, without further investigation, prove whether the colloidal silver on its own made the zone of inhibition or if the distilled water helped in the process. Here's paragraph 2. The reason why Group E had a visible inhibition zone growth is due to the extremely deadly nature of silver ions. Since they are extremely small, they can get through the E. coli cell wall easily and bind to everything they can find, like their DNA, respiratory system, and cell membrane, eventually killing the bacteria due to the bacteria's newfound inability to function. Especially since E. coli is gram-negative, its walls are thinner than other types of bacteria, which helps the colloidal silver spread and enter. Even though the colloidal silver is extremely diluted, the few spread out nanosilver particles were able to get into the bacteria, killing it and spreading it to such a significant number that it produced a clear circle or zone of inhibition around the filter circle. The ones greater than Group E's had more colloidal silver ions, so it was able to kill more bacteria. I cannot explain the control group's results at the moment, though I do think that what I observed wasn't a proper zone of inhibition due to the, like I said before, discrepancies in their appearance compared to the colloidal silver ones. This is the first part of the third paragraph. There were a lot of difficulties running this project. First, the mold growth. The mold growth impacted most of my data, and if I were to redo my experiment, I would put it in a different spot to hopefully prevent some growth and maybe get different petri dishes. Another difficulty that I experienced was measuring the zone of inhibition because, as stated before, most of the zone of inhibitions were less than 3 centimeters. The method of putting the colloidal silver onto the filter circles changed from test 1 to test 2, and that test gained higher results, so I wish I could have known that from the beginning to get more complete and accurate results. Plus, if I had higher zone of inhibitions, I wouldn't have had to eyeball some of the results that were on the smaller sides. The differences between the groups were really significant though, and that was one of the things that went better about the project. None of the constants were hard to control either. Diluting the colloidal silver is fairly easy, as well as finding visible zones of inhibition. This is part 2. As stated before, the control group was growing a zone of inhibition, though different was unexpected. I didn't end up giving test group's control group that much leverage for the zone of inhibition, but it was strange how there was something resembling a zone of inhibition, yet there were opaque, light brown dots, almost like the bacteria had clumped together in the... I'm saying this word very, strangely, but in the zone of inhibition. Another unusual thing noted was that some of the tests with colloidal silver also developed a zone of inhibition, though its appearance was like the ones in the control group, as it had 2 or even 3 rings, so I had to guess what ring to measure. One question that I'm wondering through this experiment is, how does distilled water affect bacterial growth? Here is my reference list.